Thank you, Dr. Geetamuni, for including me in this instruction course and all my co-instructors for the support in preparing this presentation. So we have been told about what an idle operation theatre is. We have been also told how to clean and sterilize it. But how do we validate that whatever cleaning and processing that we are doing is actually working out? And that is through OR surveillance. And the major goal of all the previous 3 talks and the subsequent talks is that we want to prevent a surgical site infection, and for ophthalmology that is endophthalmitis. So whenever we have a new facility and you want to start a new facility, then you do a cleaning and fumigation for at least 3 days, and each day after cleaning and fumigation, the next day you do a OR surveillance. And if the microbiological report of that comes out to be negative in all the 3 consecutive days, then you can start the OR facility. And if you have an existing facility, what you need to do is you either have a protocol that once a month or twice a month you can do a OR surveillance. We usually do it once a month. So what materials do you require? You need to have a microbiological setup or a tie-up with a microbiological lab that does these kind of processing. So you need sterile swabs, a liquid media like a brain heart infusion, solid media like blood agar, chocolate agar, Seborrheic dextrose agar, and you need biomonitors. The sample should be collected the first hour of the day when you open the OT. Immediately when you open the OT, the sample collection should be done. You need to label all the sterile swabs, and it does need not require a microbiologist. You can either train your OR sister or OR assistant to do this sample collection. And the sample should be collected from various sites. That includes the hand water source, it includes the walls, the air duct inlets, the AC duct outlet, the instrument trolley, the handles of microscope, and operation table. From the operation table, we usually take it from the head end, and the walls usually behind the wall behind where the surgeon sits. Air sampling is another important surveillance that you need to do. Commonly we do it with a settle plate method, but there's some commercial air samplers available. And how do you interpret it? There are some complicated formulae, but the simplified criteria is that in a 10 centimeter Petri dish, you should have less than 10 colonies. If you have more than that, that means there is something wrong with either the HEPA filters that are installed or something wrong with the fumigation or the sterilization processes that you are having. All the laboratory processing is done as per standard aerobic and anaerobic cultures, and those reports are given by the microbiologist. The second thing that are there are the chemical indicators. Chemical indicators are something that you can interpret yourself. There are different classes, there are 6 classes, and let me just tell you that no class is superior to the other, because all these classes of indicators are actually testing a particular process. So you need to have a class 1, that the indicator tapes. I'm sure most of you all are using it like a signal lock that is given in each load and each instrument pack. Then you need to have a class 2, a Bovidic, which also tests the steam processes and steam penetration, and that is done maybe once, recommended every day. It's frightfully expensive, so you can probably do it a weekly or fortnightly basis, and you need to do it only once a day. And lastly, you can either have a 5 or class 6 chemical sterilization indicator for each load. You need to have biomonitors also. Now they are examining and evaluating different aspects of the sterilization processes. So you have biomonitors separate for steam process and biomonitors separate for ethylene oxide process. This basically contains spores of some bacilli. And where do you place the biomonitors? You need to place it deep into your load, where the steam has to penetrate quite deeply. And after each cycle is complete, you remove the biomonitors and you crush the glass ampules that are present within the plastic ampules. And this releases the monitors into the liquid media that is present inside, and then you incubate it. If there's a color change, it indicates that there is a sterilization failure, and you need to again go back to the drawing boards to see where it has gone wrong. And as Dr. Sagar had also said, everything has to be documented. So you need to have a register or some kind of a system where you have these reports filed in for scrutiny, for monitoring how the microbiological surveillance has been there in your theatre. And it's better if you have an infection control committee that periodically evaluates these documents. These documents from the lab should go to the persons of interest, that is you. It should not be just filed away. You need to see this. you need to take actions on that, because surveillance is a dynamic process, and that is where you need to take part in it. So periodic OR surveillance is absolutely necessary, and even if you have the best of best theaters, if you are not doing periodic OR surveillance, you are not doing justice to the infrastructure that you are working in. And we all know that the operation theater is our sanctum sanctorum. That is the most precious place inside the operation theater.